Two systems, both very, very useful, both highly quantitative. The question that many people often have is which labeling system is right for you? So how I see it is, if you are in a research group or you are working on a project where you are interested in perhaps a large number of genes, and the genes that you are interested in are typically changing quite frequently, then you might want to go for a technique like CyberGreen, where you are not restricted to sequence-specific amplification. On the other hand, if you are a group where your sole focus is a handful of genes and you want to be really, really highly quantitative and highly specific with those genes, then you might decide to invest in the fluorescent quencher probes. But at the end of the day, it's a weighing up of the advantages and disadvantages, and it often comes down to the cost. So let's now look at the advantages and disadvantages of each. So if we start off with CyberGreen, one of the major advantages, of course, is that it is so flexible because it can amplify any double-stranded DNA sequence. On the flip side, this increases the risk of false positives, and that is because CyberGreen can also bind things like primer dimers, and it can also bind off targets, and it's difficult to discriminate between these things during the PCR reaction. On the other hand, another benefit is that CyberGreen is very cost effective because you don't need to go through the expensive process of generating a probe, and this may reduce your assay and running costs. But in order to get the best out of something like CyberGreen, you will need to do a lot of optimization on your primers to make sure that they do not amplify anything that is off target. And this in itself can be time consuming and cumbersome. And that's where you need to take into consideration, well, time is money too. So it's not straightforward. However, CyberGreen can be straightforward if you're very lucky and if you're working with genes which have very specific sequences for primers. Then you can just carry on with CyberGreen without worrying about the challenging probe design. But you will always need to run a melting curve at the end. And then on to fluorescent quencher probes. Well, the reason they were generated in the first place or developed was because of their increased specificity. So it's a single probe, single target scenario. One of the beauties, actually, of fluorescent quencher probes is that it's possible to carry out multiplex reactions. So what I mean by this is, because probes can be labeled with distinguishable dyes, it's actually possible to have two or more probes in the one reaction. And in this way, you can simultaneously detect the expression of many different genes. And one of the attractions of this is that you rule out the variation that you might get between different PCR reactions. And in ways, it can be time and money saving to have a probe if you are working in on a very focused number of genes, and you have a well-developed probe. Then you will maybe in the long term save time and money because you won't need to post-run melting curves and spend a lot of time optimizing primers all of the time. But let's not make it too simple because probe design can be challenging, and I will not really focus on probe design today. But I believe that if you are interested in buying a probe, many of the companies who generate probes will be only too happy to assist you. Probes can also be expensive because you need a single probe for a single gene, and sometimes they can have the downside in that they can be inflexible and can lead to false negatives. And this could happen if you were analyzing a gene which had many different splice variants. It would be a good idea to try and find out beforehand if your probe identified splice variants or not.
To view the full video of this and all of our other webinars for bioscientists at the bench, please visit bitesizebio.com/webinars.